concept of genomics and fungal identifications. Okay. So lastly, right? So we found the problems, right? Because typically in fungal identifications, right, we rely a lot on morphology, right? So that kind of relates to the physiology that we discussed, right? Depending on how they reproduce, depending on how they grow, right? But in reality, right, we actually have to use other kind of technique like DNA, right, or phylogenetic trees to help, okay, using their kind of DNA informations. But if you take a look at this study, this was from 2017, around 7, 8 years ago. They actually survey, right, some of these study. Oh, this is a fungal study, right? So unlike bacteria, uh, fungal study they rely a lot on morphology. So 31%, which is kind of crazy, right? This is just not even 10 years ago. Okay. But if you compare to bacterial, right, which is probably a lot of them using the DNA for identification already because it's much simpler, right? You use 16S, right, kind of give the results, but fungi might not, right? Okay, so 28%, right, don't even tell you which identification procedure that they use. So almost 60%, you kind of rely on morphology and something that you don't even know. Okay, and if you take a look at molecular, right, using DNA, it's roughly around 50%, from 40%, not 50%. So it's a lot less, right? This is not even 10 years ago. But as of right now, it's a lot better than 10 years ago. But still, right? We still rely a lot on morphologies, okay? Because it's very different from the bacterial, right? You cannot use much of morphology, but for this one, you rely a lot on their structures, okay? Now, let's identify why this morphology is so challenging, right? So first of all, even though we see a lot of their structures like sexual development, right, and also those kind of structures that they produce depending on how they grow, how they reproduce, but basically a lot of these have limitations in the number of characters that we can use. Okay, all right. Secondly, we talk about conversion and evolutions. Remember, last time we talked about one example of conversion and evolutions. What does it mean? Conversion and evolutions.
Into one.
What does it mean? Into what?
Not the same access but the
Right. Okay, so there's some evolution happening, right? They have very similar functions of morphology, physiology, whatever, but they not really. They don't have the same existence. So that's also being problematic in fungi. Because some of these, you can identify them using morphology. It's very clear. But later you found that you also have very similar species that have very similar structures. But later you found that they're not coming from the same system. Okay? Right? And morphology in fungi, a lot of them, you can't really use morphology to identify as species level. Very hard to do. And a lot of these fungi, you can't culture them. Okay? So it's very hard, even harder. So take a look at the next slide. Okay, take a look at this one. So this is always my kind of examples that I will typically use to show a conversion evolution in fungi. This is example of, well, the mushroom. So two of this, one of them is actually edible. The other one, well, technically you can eat it, but it will affect your body. So you should not eat it. Alright? So on the left side, on the right side, if you find this in the forest or in the soils, right, you might think it's one of this. But once you put it side by side, you see that the color is just a little bit different, right? But in the actual future, you might not be able to differentiate them. And you may think example, you lost in the jungle for quite some time, right? And then you see, oh, I found this mushroom. John told me you can eat it, right? But it looked very similar, right? So that's going to be very hard. So it's like 50-50 chance, right? So you take a look at this picture. Oh, this one is a little bit less pink than the other one. Probably okay, right? But it might not be real because in environment, right, these pigment might actually be different based on that data. Okay, so morphology, right? They might look very similar, but it's actually different species, different groups, right? They are actually different orders. Okay, so that means the materials metabolized and used will also be different. So their physiology is actually quite different. Okay, so that's the reasons why we have to have these combinations of identifications. So that means when you if you get lost in the jungles, so don't eat much food. That's not your go-to friends, right? Because you have higher chance of getting toxins or probably get high, right? From this okay? And you may do things that you should not do. Okay, so find something else to eat, right? Not the much food. Now, the reason that I brought this up because in your generations, things are getting cheaper, right? You probably have seen a lot of this presentation using this number here, right? In the past, if you go back, 15 years ago or so, the price of the sequencing is way high. But in your generations, it's much cheaper. And also, if you take a look at these timeline, so the top right over here, the time it takes for generating your sequence is much shorter and you get more data. Okay? So this is what we call parallel sequencing. So in the past you sequence probably one at a time, right? Now you can pull the sample, right? Now you can sequence a lot of these at the same time. Okay, now the question is, how would you do it? Right? You take a look at the chromosome, you take a look at the genetics. So in bacteria, it's very simple, right? 16 is probably be enough, right? Or indications. But for fungi, it's very different. It's depend on, the group of fungi that you want to study. Some of them, you might be able to use this ITS1, ITS2 for identify at species level. But in some group, you might actually have to use other things like 18S or 28S in combinations. It really depends. But typically, in general, based on the published data, right, of these fungi, you can typically use ITS for universal barcode for fungi. Okay? Right. And obviously, you probably have to use this PCR, right, to identify, to make more copy of this, and then later, right, you have these DNA and then you identify them, right, using the database. For example, you can use NCBI by using BLAST or whatever. Now, what I want to show you is that, well, the fungi that you want to identify, we actually have different databases. So it might be wise to take a look at these databases. For those of you who work with fungi, might have to take a look at some of these fungi in your project. So for example, if you say that you want to just identify the typical fungi, obviously all these databases that I'm showing you, they all have ITS. Okay, so you can typically use ITS and you can use that for identifying any of these databases. But if you want to be more specific, okay, for example, this channel over here, which is focus on the fungi and in human animal, you might want to use, for example, ITS and not other things. Okay? Right? But you may also use, for example, QBAC here. They actually have other kinds of genes that you can use for identifications. Okay? Right, so this really depends on what you want to study, right? But you also probably want to take a look at how often they update the database. Okay, so suggestions, even though this is the last on the list, this one here, the Unite database, is probably the most outdated fungal database that we can use. And they also have very similar kind of platform, which is similar to the blast search here. For example, you can get the DNA sequence, right? And you can blast search database on NCBI. Okay? On Unite database, they have very similar platform like this. You can blast search on the database, and then it will show you pretty much the same results. But the key thing is NCBI. Anyone could input or publish their sequence. So your sequence might match to their, whatever sequence that I publish, but the species that I provide to the database might not be correct. Okay, so that's kind of one of the downfall of the public database. But for Unite database, they are researchers who take a look at the data that got submitted to the system, and they will review these database to make sure that the data that we put in is actually makes sense. Okay, because you might find that in NCBI sometimes you match, right, your sequence right, to the database, and then kind of the measure of the list may say, oh, this is all Bacillus, and somehow one of them show you this is E. coli, right? So you might find that happening in NCBI. Okay, so make sure that when you take a look at those databases, just try to be curious about the results that you get. So don't just say that, okay, this is what I found, and then I'll just report this. Okay? Alright, so hopefully what I show you here is that there's a lot of different databases that you can use. Okay? So if you're not sure how to use it, just go to several of these and then just try to match them, right, using your sequence. And if they all get the same results, then it's probably correct. If not, then you might have to think about, is it the database itself? Is it your sequence? Right, so just think about it. Okay, now, just to summarize all of this, as of right now for fungal identifications, these are the typical things that you can use. You can use ITS, right, depending on whatever regions that you use, but basically all the databases that I showed you, they all have ITS, right, as a kind of reference for you. Right? You can use Sanger sequencing, or you can use the new next generation sequencing, parallel sequencing, and then you can search them on these reference sequence depending on the database. And additionally, depending on right the result that you get, you may use these additional keys right for identifications. Okay. Now what I want you to compare is we have the blast search. Right? Think about it. That's kind of the original, right, kind of sequencing that you probably think of when I say using sequencing for identifications. But if you think about it, so you probably have used NCBI before. If you get the sequence, okay, from the sequencer, okay, and then you try to match them to the database, how long would it take you? So you put them in the website. How long? You probably have tried it. How long did it take you before you get the results? Is it like half an hour, or just 30 seconds, 1 minute? Have you tried it? Yes, you have. How long did it take you? Short time or long time? Quite short time, right? Okay, but now think about this. Okay, here what I want to show you. Sorry about my handwriting again. Okay, so basically when you compare with the next generation sequencing, like I said, this is Sanger sequencing versus parallel sequencing. So you compare the sequencing platform that you can sequence many things at the same time. So for Sanger sequencing, you get many copies of identical sequence. So that means the sequence that you typically put in NCBI, this is just one sequence, right? One long chain of the DNA that you get. But in NGS, you get millions of sequences, which are not identical. Some of them may be identical, but a lot of them are not. Okay? Alright, now the key thing is, for these one sequence, you probably take like 30 seconds or so, right, to match the database. think about those million sequence. If you have to use the blast search, how long would it take you? 30 seconds on 20 million sequence? Around 19 weeks. Think about it. Right? So if we still using that same technique, right? Your master degree will take 19 years. That's not a good thing, right? Okay, so in your generations, you should at least understand the technique that you have. If it can save you time, but it might be a little bit kind of more expensive, right? Then use that one. It will only take like hours to identify, if you have example 100 or even 200 samples, okay? But it really depends on the questions. Okay, you can't just do FGS on all the samples, right? But if you have a lot of samples that you can actually pull them or using very similar procedures of sequencing, then you can use that one. It will save you a lot of time. But if you only have, for example, you only have one isolate, you have 10 isolates or whatever, right? This will probably be fine, right? If you think about, if you have only 10 sequence, right, you multiply that, it's only take you like 5 minutes or so, okay? But if you have like millions of sequence, right, you can't do this, right? It's harder. Okay, right, so the last part, I would like to talk about this method of coding a little bit. Well, it's kind of the technology that everyone can use now, very easy to do. Just to give you an idea, since you are probably going to write your proposals very soon, some of you might have already started. So this is one technique that if you want to study the microbial communities, okay? So the key thing that you actually need is the barcode for that particular organism. For example, if I want to study fungi, I can just use ITS. Or if I want to be more specific, okay, I can use adenine bacterials or something that's specific to the group that I want. Okay, so this is really depends on the organisms that you want. You can use 16S or bacteria, or you can be more specific if you want to study just one group of fungi. The bacteria. Instead of using 16S, you may use other genes. Okay, and you can still trying to find their complex communities and their diversity. The key concept is that when you take a look at these kind of sequencing, things are much more complex than Sankey sequencing. Sankey sequencing, you get, a lot of sequence, but they are all identical, right? So that means you have one sequence. But for this one, you have, think about of these as puzzles, right? You have like one big picture or something, right? And then you have these different puzzles that might be coming from different pictures. Okay. The key thing is how do you differentiate them parts? This is not about identifying fungi, not yet, right? What you want is you want to be able to separate them. And in order to use this technique, you have to be able to separate these 20 million reads, right, into those kind of 200 or 100 samples that you have, okay? And secondly, after you separate them, you can tell which one apart by using taxonomy identification, using the database that I was telling you earlier, okay? But not using the blast because that might take you 19 years, okay? All right, so how do we do this? Well, the first one is very simple, okay? This rely on your wet lab. So when you do those PCR, you can actually adding this little tag to your sequence before you put it into the sequencer, okay? For example, here, you see that each of these puzzles, right, you have this little contour here that have different colors. So think about adding additional sequence to your PCR, right? These additional sequence will be unique to particular samples. So for example, in sample 1, you have these orange colors. So that sequence will be unique to sample 1, okay? And that will be the same for sample 2 and sample 3, they will have unique sequence. So after you get that sequence, after you get that puzzle, right, it seems like you have these indications of where it's coming from, okay? Now the second question is, these puzzles, after you separate them out into samples, how could you identify them? Well, take a look at this. This is puzzle of the animals. If you know one corner of these puzzles, you can't really guess which animal they are, right? So it might be coming from something like this. So think about this as, the microbe itself, right? You don't need to know the whole genome of that particular organisms, but if you know the right part of their genomes, you can compare between them, and then you will know which microbes they are. So this is kind of the underlying mechanisms of how these parallel sequences work, but in reality, they're actually much more complex. So this is just to give you an idea of what you can use as of right now. Okay. There's actually other sequencing technique that you can use. As of right now, the third generation sequencing, you get more data, right? You also getting longer sequence comparing to this metaboloically. You don't require amplifications, you don't require PCR, okay? But the cost actually much higher. So if you're rich enough, yes, go ahead for it. If not, this is probably enough for many other patients. Okay? But if you want to take a look at the genes like the mating type, mating locus, you probably require longer memories. So you probably require the new generations, even right, the third generations of these NGS. Okay? So, but if you don't want to know anything about their genes, you just want to identify what they are, right? This is probably enough for you. Okay? So take a look at your research questions and use the right technique for your project. Okay, so for these kind of questions at the end, this is what you get. So this is kind of in the part now, we call them operational taxonomy unit. This is kind of representations of the species of each of those sequences you get. Okay, so in one system, for example, in soil, you may have thousands of these different fungi or bacteria in the soils, and then you can differentiate that, right, into a sample, like how much you have of each of that species. Okay, all right, that's actually it for today. So hopefully you see, there are a lot of things that rely on fungal genetics and pharmacogenetics, right, ranging from how they reproduce to how we identify them. So in general, from these two lectures, hopefully you see that their morphology, physiology, their behavior, their sex reproductions rely a lot on what's happening within their genes and the regulations. So you might have to use a lot of techniques in order to study it. Okay, so now the last thing I would like to show you, this is your assignment. So we have around 30 minutes left, but these lectures, this research talk is only around 20 minutes. Okay, basically, Professor Steich, he presents the coccidioides, which is one of the key fungi that creating problems in the United States. So he's take a look at these genomes, right, and compare between these different species that they find. So I want you to take a look at these research and hopefully relate it back to what we learned in class. But it's probably going to be much more complicated than what we learned in the class because we only have 2 lectures. I posted, it should be available in my course group by now because I scheduled the post at 10:30, so you probably see it. So these exact same descriptions. So you are graduate students. I'm not asking you to summarize these lectures. What I'm asking you is that I want you to take notes, right, of these research talk that you see, and then do 4 things. Okay. So the first one is try to find the personal connections, either the knowledge that you get from this class or from your research or whatever. Is there anything that related to your experience, to your knowledge? Why does it matter to you personally? Okay. So that means these will be different for each of you. Okay, this is your personal connections. Okay. Second, critical analysis. Okay. So I'm asking you to take a step further, right? To think about the research that you just watched. Identify what is the key concept, that is the strength of the research, the limitations. Okay? Right? So if Professor Steich kind of talk about this, do not just repeat what he said. You have to reflect on what he said. based on the video that you will watch, it's not going to be direct. So you have to find this on Google. Okay? And then third, the applications. Okay? So from these research that you watch, trying to apply. Okay? Whatever you see, whatever you understand from that lectures, trying to think about the application of these research. Okay? Right. And then lastly, discussion question. Okay. So based on whatever you write, the final part, I want it to be your kind of final discussions. Okay, just one key thing, one thoughtful questions, okay, based on these research. Okay, if you were there, okay, what would be the questions that you would like to ask Professor Sledge? Okay? Right. Okay, so think about all this. You may use AI to help you, assess you, but do not copy everything. Just rely on your critical thinking. Okay. Right? So if you just copy everything, I will not accept that. Okay? Right? Even though I can't tell 100% whether they are being generated from AI or not, but I will try to see. There's some keywords. You probably have seen it because there's some keywords that AI typically use. So if you use AI as in your assignments, try to think about that, whether or not they answer these questions. Okay? I'm trying to make this assignment so that it's more related to yourselves. Okay? So hopefully that's not too hard. You can use AI to help you summarize the video or whatever, but then when you write this assignment, I'm asking you only 2 to 3 pages, so probably like 4 paragraphs or so, right? Not too much to kind of reflex this. So maybe 1 paragraph to your personal connections, 1 paragraph, the second part, third, and fourth. Right? So probably like 2 pages or so. Okay? Alright. So these will be till next week. So you have 1 week to do this.